So I welcome you to the next class, that is part 3 or chapter 3 you can call it, of embryology, where I shall be discussing three important topics. The first one is a reproductive system. And following this, we shall be discussing the gametogenesis. And then we shall be talking about the ovarian cycle and the menstrual cycle. So in my topic of today's discussion, the important highlights are, the first thing is we shall be discussing about the process of reproduction. The reproduction is nothing but it is a process of formation of a living organism. A new human being is manufactured because of the reproduction is a property that is seen in higher animals, and because of the presence of dimorphic gametes and because of presence of sex organs, so the process of reproduction is carried out so that the offspring is continued. In this process, you need to understand that the sex organs are very important because they are secreting gametes. These gametes are nothing but the sex cells. The gamete in male, you are calling it as a spermatozoa. We'll discuss how is this spermatozoa formed. Spermatogenesis we'll discuss. Spermatozoa is produced in testis, and in females the gametes are called as ova. Ovum is singular, ova is plural, and produced by ovaries. Ovary produces cells called as ova. Testis produces cells called as spermatozoa. When spermatozoa and ova combine together, they result in formation of a zygote. This zygote is responsible for formation of a new human being subsequently, and that study prenatally we are calling it as embryology. So this is the very this is the very beginning of embryology, right? So the process of formation of gametes, we call it gametogenesis. The process of formation of gametes, you are calling it as gametogenesis. What are the two types of gametes? I'm sorry. Excuse me. What are the two types of gametes? We have got spermatozoa and ova. Production of spermatozoa we are calling it as spermatogenesis. Production of ova we are calling it as oogenesis. So subsequently you can divide gametogenesis into spermatogenesis and oogenesis. Oogenesis. Hope you are following this. The spermatozoa are derived from cells called as spermatids. Because they are round in shape, we are calling it as round spermatids. Spermatids. spermatogenesis is different from spermiogenesis? It's an important entrance question. What is spermiogenesis? Spermiogenesis is a process of conversion of a typical spermatid into a specialized spermatozoon. So if this spermatid gets converted to a specialized spermatozoon, this process is called as spermiogenesis. The process of formation of sperms, the overall sperms, you are calling it as a spermatogenesis, whereas the small process, the very process of conversion of spermatids to spermatozoon, you are calling it as spermiogenesis. Next comes the understanding of oogenesis. Oogenesis is nothing but the formation of ova, which occurs in the ovaries. So say suppose this is an ovary. Ovary has got multiple primordial germ cells. One of these typically grows in a month to form a mature oocyte, mature follicle, which pops out an ovum outside, and then the mature follicle you are calling it as a Graafian follicle. I'll discuss that each and every point today. Graafian follicle we call it. Every month one ovum is released out. One ovum is released out every month. Like that, in a female, the primary germ cells, so these are the primary germ cells or primordial germ cells, primary oocytes, which gets transformed into secondary oocyte. Then secondary oocyte undergoes fertilization. I'll discuss that, how fertilization is occurring. And this is throughout the menstruation cycle. The physiology of menstruation, I discussed this point in my physiology classes. Again, I'll discuss much more details in gynecology chapter, gynecology topic, and in that chapter is the infertility chapter, infertility. I'll discuss that further. Here also I'll discuss the most important aspects related to embryology. Then that is just before onset of menstruation. What are the changes occurring in the uterus, changes occurring in the innermost lining of the uterus, changes occurring in the ovaries. These are the ovaries attached, ovarian ligament. All right, so something like this. So we'll discuss all those points today. This is the summary of today's class. We'll begin our discussion. So as I already told you, the germ cells are very, very important component of gonads. Testis is a gonad, ovary is a gonad, respectively in male and females. So gametes or germ cells are important component of these gonads. The male gametes are spermatozoa and the female gametes are ovum. All right. The growth and development of a fertilized egg occurs in the female reproductive system for a period of 10 lunar months. 10 lunar months. We call this lady as a pregnant woman, and immediately following delivery, then changes occur in the mother's reproductive system, and a lot of milk is secreted in the mammary glands, and this period you are calling it as a lactation. All right. So now let us discuss how the male reproductive system is functional. The gonads in the male reproductive system. We'll start our discussion with spermatogenesis, but before that we'll try to understand what is the male reproductive system overall. We all know that it is the testis. Testis is a plural. Testis is a singular. So you can write either the way if you're talking singular or plural. So the gonads are produced, these gonads produce male sex cells and secrete male hormone called as testosterone. The testis, both the testis right and left, it has got duct system. Now this duct system composed of epididymis, the vas deferens, the ejaculatory ducts and the urethra. which connect to the penile urethra, penis. Remember, the duct system opens up in the penile urethra. Penis is a copulatory organ. It assists in storage, transport of these spermatozoa. It assists in storage and transport of these spermatozoa. Spermatozoa, right? All right. Give me 5 minutes, I'll be back. Sorry for that break. So welcome back. I welcome you to the further discussion of male reproductive system. So here what happens is there is a the muscular copulatory organ which is responsible for the copulatory organ which is responsible for reproduction is penis. So from it, what is the function of penis is it helps in transport of storage, it helps in transport and maturation of the male sex cells, that is the spermatozoa. We do have accessory sex glands called as seminal vesicles and the prostate gland. These are responsible for secretions which assist the movement of sperms. Through these secretions the sperms get nutrition and transport. So nutrition and transport, seminal vesicle secretions and prostatic secretions, they together form what is known as the semen. This the semen is nothing but the secretions of seminal vesicles, the secretions of prostate gland along with the spermatozoa. One important point I'll tell you that the seminal vesicle secretions are acidic in nature and prostate gland secretions are alkaline in nature. Acidic in nature and alkaline in nature. Alright. Testis is located in the scrotum and it protects the scrotum, basically hanging down, the testis hang down away from the abdomen so that the high body temperature is actually preventing the growth of spermatozoa. It prevents the growth of spermatozoa. It should not be exposed to high temperature. That's the reason why if somebody is wearing a tight trouser for a very long time or a tight undergarments for a very long time, then such people have a risk of spermatozoa development disorders. All right. So now let us try to understand what are the stages of spermatogenesis. The stages of spermatogenesis, you can divide it into prenatal, birth, at the time of birth, and puberty. And after that, as the child attains puberty, after puberty, the reproductive years, there are few changes that are occurring. All right, you can divide it into multiple stages like this, depending on the age of the individual. So if you see the prenatal life, in the prenatal life, we have what is known as the primordial germ cells. The primordial germ cells which are present in the yolk sac. These primordial germ cells multiply during the prenatal life, that is during the embryonic life. A lot of multiplication occurs, and this simple multiplication, it will give rise to what is known as sex cord cells. Sex cord cells also called as spermatogonia. Formation of spermatogonia. Now, this occurs at birth, and here the spermatogonia undergo resting phase. Spermatogonia undergo resting phase. After this, what happens is till puberty, the resting phase of spermatogonia is continued. After that, the spermatogonia in the seminiferous tubules at the time of puberty, in the seminiferous tubules, that is a duct system, they undergo multiple divisions. They divide into what is known as spermatogonia type A or type 1. This spermatogonia type A undergoes 4 successive multiplications, results in formation of type B. Four divisions or four multiplications to form type B spermatogonia. This type B spermatogonia undergoes mitotic division. Yesterday we read what is mitosis, what is meiosis. So it undergoes mitotic division to form what is known as primary oocyte. So primary oocyte is a product of mitotic division of type B spermatogonium. In mitosis, what is the important feature of mitosis? How many cells are formed after mitosis? 2 cells are formed. So here 2 primary oocytes are formed. In case of mitosis, the daughter chromosome is same as parent chromosome. That is, it is diploid. Is it not? It is diploid. Division of nucleus does not occur. Alright. So this primary oocyte now comes important step, that is it undergoes first meiotic division. First meiotic division results in formation of secondary spermatocyte. Oh, I'm sorry, it is not primary oocyte, it is spermatocyte. I'm really sorry. Sorry for the mistake. So it is primary spermatocyte. Oocyte is in females. I repeat again because of the mistake. So spermatogonium in the seminiferous tubule undergoes first division to cause to become type A spermatogonium. Type A spermatogonium undergoes 4 multiplications or 4 divisions to form type B spermatogonium. Type B spermatogonium undergoes a mitotic division, the first mitotic division, and results in formation of a primary spermatocyte. The primary spermatocyte has got diploid chromosomes. The primary spermatocyte daughter cells are 2 in number with diploid chromosomes. After that, what happens is it undergoes first meiotic division to form what is known as secondary spermatocyte. Okay, secondary spermatocyte. We all know that the product of first meiotic division will result in formation of 2 daughter cells each. So 2 cells of primary spermatocyte, again 2 daughter cells each, we get 4 number of secondary oocyte. And what is the condition of the nucleus? Is it diploid or haploid? Here it is haploid, is it not? Because meiotic division undergoes reduction in the number of chromosomes, exactly half. The number of chromosomes is reduced to half after first meiotic division. Alright, this secondary spermatocyte undergoes second meiotic division, meiosis 2. Second meiotic division to form spermatids. These spermatids are 8 in number, and obviously they are haploid. And each spermatid undergoes division, further metamorphosis we call it. Change in the morphology is metamorphosis. Change in the morphology. It undergoes change in the morphology to result in formation of what? The final end product, that is a spermatozoa. I told you that the formation of spermatozoa from the spermatids, this process is called as what? Spermiogenesis. And this entire flowchart is called as spermatogenesis. So spermatogenesis includes from the primordial germ cell in the prenatal life in the yolk sac, primordial germ cell undergoes multiplications to form spermatogonia. Spermatogonia present in the sex cord cells. The sex cord cells also called as spermatogonia. At the time of birth, it undergoes resting phase. At the time of puberty, what happens is the spermatogonia and the seminiferous tubule undergoes changes to form spermatogonia type A. The type A spermatogonium undergoes 4 multiplication divisions, 4 successive divisions to form spermatogonia type B. The spermatogonia type B is termed gets converted to primary spermatocyte. The primary spermatocyte is formed by mitotic division. Mitosis results in formation of primary spermatocyte. Primary spermatocyte is diploid in number. Primary spermatocyte, there are 2 cells formed from each spermatogonium. There are 2 cells formed, the 2 number of cells are formed in each spermatogonium, and we are calling it as a primary spermatocyte. It is a product of meiotic division. After mitotic division. After mitosis, it undergoes first meiosis. The important property of first meiotic division is 2 cells are formed from each, two daughter cells are formed from each parent cell, which is undergoing meiosis 1, and then the number of chromosome is reduced to half. So the diploid chromosome becomes haploid, and the product now formed is termed as the secondary spermatocyte. The secondary spermatocyte. The product formed is termed as the secondary spermatocyte. The secondary spermatocyte are 4 in number because 2 primary spermatocytes are undergoing meiotic division to form 4 number of haploid spermatocytes. These spermatocytes undergo second meiotic division. Meiosis results in formation of 8 spermatids, which are obviously haploid. Then from spermatids, it results in formation of what is known as spermatozoa, which are 8 in number. So we all know that from spermatid to spermatozoa, it is called as what? It is called as spermiogenesis. Alright. So the formation of primary oocyte to the formation of spermatids. The formation of primary oocyte to the formation of spermatids. This is termed as meiotic phase of reproductive system, male reproductive system, meiotic phase. So the first meiotic division results in formation of secondary spermatocyte. The second meiotic division results in formation of spermatids. This is called as meiotic phase. All right. The process of change of spermatogonium from the puberty, the process of change of spermatogonium to primary spermatocyte. This is called as spermatocytosis. Spermatocytosis. So we have got 4 terminologies here. First terminology that you should understand is what is spermatogenesis. What is spermatogenesis is first terminology that you need to understand. Second terminology that you need to understand is what is spermatocytosis. Spermatocytosis is maturation of the spermatogonia into primary oocyte. Next, what you will have to understand is the meiotic phase includes from primary oocyte, from primary spermatocyte to spermatid. We are calling it as a meiosis, which includes meiosis 1 and meiosis 2. After that, from spermatids to spermatozoa, you are calling it as spermiogenesis. Spermiogenesis. All right. So please remember, each testis we have got seminiferous tubules. Each testis we have got so I told you the seminiferous tubules are responsible for change of spermatogonia, is it not? So these seminiferous tubules are nothing but in each testis we have got multiple tubules like this called as seminiferous tubules. These seminiferous tubules, they form what is known as exocrine part of testis. They have got in between, they have got interstitial cells. Interstitium is full of certain cells. We call it interstitial cells of the Leydig cells. These are the seminiferous tubules. Blue color ones are seminiferous tubules. Then the interstitial cells of the Leydig, we call it, which form the endocrine part of the testis. So if they ask you what is the endocrine part of the testis, it is the Leydig cell. If they ask you what is the exocrine part of the testis, it is the seminiferous tubule. It is the seminiferous tubule. The spermatozoa are produced in the seminiferous tubule by a process called as spermatogenesis. The seminiferous tubules are structural and functional unit of the testis. Please remember this point. Seminiferous tubules are the structural and functional unit of the kidney of the testis. They are the structural and functional unit of the testis. All right. Each seminiferous tubule is around 2 feet in length. They are coiled in that 70 to 80 centimeters of small area of the testis, 2 feet in length. All right. You can understand how much coiling of the seminiferous tubule is occurring. These seminiferous tubules, they end up, so we have got seminiferous tubules like this. So this is the testis, say suppose. The seminiferous tubules, they line up and end into a complex collection of ducts. We are calling it as a rete testis. Rete testis. This rete testis undergoes again multiple divisions to form mediastinum of the testis. From there, so we have got again multiple tubules here which open up in the vas deferens. So spermatids, the spermatogonium or spermatogenesis is occurring here. From here the spermatozoa are traveling along all these complex ducts and reaching the vas deferens. All right, reaching the vas deferens. So near the head of the testis we have got multiple coiled tubules. We are calling it as epididymis. Epididymis. This epididymis consists of multiple tubules like this, which are responsible for storage and transport of the sperms. The outermost covering of the testis, you are calling it as a tunica vaginalis. The tunica vaginalis. The inner lining of the testis, which consists of each lobules like this. This is tunica albuginea. So outermost lining is tunica vaginalis. Inner lining is tunica albuginea. I'll discuss this when I am discussing reproductive system anatomy. This is just a vertical section of the testis to understand spermatogenesis. So please remember that I already told you that the spermatogenic cells, they have got different names in the development. The stem cells are nothing but the spermatogonia. So if you see this flow chart, spermatogonia or the sex cord cells, these are the stem cells of the testis. I told you what are the stem cells. I told you what are the different types of stem cells in the last class. So they develop from primordial germ cells. The spermatozoa which are released in the lumen of the seminiferous tubules, and in the seminiferous tubules we know that lot of changes occur. That is, the spermatogonia undergo changes from type A to type B. This process is called as spermatocytosis. This is occurring at puberty. In the early reproductive life and throughout the rest of the life, what happens is the meiotic division occurs and spermiogenesis occurs, results in formation of the spermatozoa. By meiotic division, what is formed? The primary spermatocyte gets converted to the secondary spermatocyte. Give me 2 minutes break for azan. All right. Welcome back. So I was discussing what happens, what are the products of primary meiotic division. What happens is primary spermatocyte undergoes division and to form secondary spermatocyte. The secondary spermatocyte again becomes spermatids. So this process is called as meiosis. Please remember that the seminiferous tubules have got spermatogenic cells and supporting cells. The seminiferous tubules, again I am talking about these tubules, the coil tubules, all right. Again I am talking about this seminiferous tubules. So the seminiferous tubule, so this is the seminiferous tubule, all right. The seminiferous tubule has got two types of cells. It has got two types of cells. One is the spermatogenic cells, which are forming the spermatogenesis. The other type of cell you are calling it as a Sertoli cell. What is the importance of Sertoli cell? Sertoli cell is attached to the basement membrane of the seminiferous tubule, and it has got an elaborate system of thin processes, very small processes into the lumen of the seminiferous tubule. And Sertoli cells actually they give nutrition to the developing spermatozoa. And there is one important hormone secreted by the Sertoli cells. It's a question for you. It is inhibin. Outside the seminiferous tubules, we have got the interstitial cells. Interstitial cells are called as the Leydig cells. Isn't it? Just now I told you. Leydig cells. Leydig cells of testis are endocrine in function. They are secreting an hormone called as testosterone. Under the influence of pituitary gland, this hormone is functioning. The Leydig cells are rich in two important. They are rich in vitamin C. In fact, vitamin C is responsible for the metabolism of testosterone, and they are rich in phospholipids and fats. They are rich in fat, they are rich in phospholipids, and the function of testosterone, you all know that it stimulates the production of the external genitalia of a male, all right. So function aspect, we have already discussed testosterone, how does it act and all in my physiology class. So for now, please remember, this is all about the spermatogenesis. I repeat again very quickly. So primordial germ cells in the prenatal life undergo multiplications to form the stem cells, that is the sex cord cells, also called as spermatogonia. Now the growth stops till the puberty. Till the puberty, there is no growth occurring. After that, what happens is the spermatogonia undergoes, at puberty, the spermatogonia undergoes division and into formation of type A cells, then type B cells. This process you are calling as spermatocytosis till the formation of the primary spermatocyte. From primary spermatocyte to spermatids, you are calling it as meiosis, because meiosis 1 occurs, meiosis 2 occurs, and formation of a haploid spermatids occurs. Spermatids are round in shape. They have to become elaborate, and the typical head and tail and all this results in the process called as metamorphosis, and the stage is called as spermiogenesis. The stage is called as spermiogenesis, results in formation of 8 numbers of each secondary spermatocyte, give rise to 8 spermatids and 8 spermatozoa. 8 spermatids and 8 spermatozoa. The process is spermiogenesis. Hope you have understood this point. Now let us talk about the female reproductive system. So many of the students find female reproductive system to be slightly complex, thinking that owing to the fact that they are not properly prepared, they don't properly prepare for the topic, and simply they say that, sir, this topic is very tough for us, you please explain it properly. So I again and again tell them that I'll explain you, but you have to revise it. After the video is over, you have to sit down and try to recollect what I have told, so that you understand the female reproductive system properly. In the female reproductive system, we have got a uterus with fallopian tubes. These fallopian tubes help in collection of the ova. We have got ovary attached like this to the ovarian ligament in the broad ligament of the uterus. From here what happens is an ovum comes out every month. It is taken by the fallopian tube. In the fallopian tube, fertilization occurs, zygote is formed, zygote gets transported. Slowly it moves into the fundus of the uterus. It gets implanted to form what is known as the embryo. This is what is the study of embryology. So here we will discuss two aspects. The first aspect we will discuss the ovarian cycle. Second aspect we will discuss the fertilization. Second aspect we will discuss the fertilization. In the ovarian cycle we will discuss ovary is a gonad. So in the females the gonad is ovary. Now this ovary is responsible for formation of certain follicles. These follicles are secreting every month one ovum which undergoes ovulation. Every month this is happening, whether the sperms are present in the genital tract or not. So let us try to understand this stage of oogenesis. Previously we discussed what is known as spermatogenesis. Now we are talking about oogenesis. Oogenesis, the flow chart we will try to understand. So here also what happens is the story starts with the primordial germ cells. Here also in the prenatal life the story starts with the primordial germ cells. The primordial germ cells undergo mitosis to form what is known as oogonia. So the chromosome type is 44XX here. The oogonia undergoes multiple divisions, mitosis again, to form what is known as primary oocyte. This primary oocyte now it undergoes first meiosis. To form the primordial follicle. Follicle means what? Follicle means a cyst. In medicine, follicle means a cyst. All right. So a small cyst is formed. Cyst like a collection of cells is formed, and you're calling it as a primordial follicle. The primordial follicle now, what happens is, as the meiosis is going on, what are the stages of meiosis? Yesterday I told you prophase, metaphase, anaphase, telophase. Is it not? Prophase again divided into leptotene, zygotene, diplotene, and pachytene, if you remember my last class. If you remember my yesterday's class, I'll show you. So this is what I told you in my yesterday's class. The leptotene, zygotene, pachytene, and diplotene. Leptotene, zygotene, pachytene, and diplotene. Do you remember this? So what happens is, once meiosis is occurring, primary oocyte, primordial follicle is formed, there is arrest of meiosis. There is arrest of meiosis in prophase 1. This is very, very important point for your entrance exams. This is a very, very important point for your entrance exams. So I repeat again, what happens to primary oocytes is that there is arrest of meiosis. So the primordial follicle which is formed here, it is undergoing arrest of meiosis. Arrest of meiosis, that is in the diplotene stage of prophase 1. Prophase 1 arrest occurs, meiosis gets arrested, that is there is stoppage of division now. So arrest of meiosis 1, that is especially in the prophase 1 phase. Now what happens is, there is atresia of primordial follicles. There is growth of only 5 to 15 primordial follicles into secondary follicles, and that become mature Graafian follicle. All right. So what happens here is the primordial follicles which are arrested, they are around 40,000. So the arrest means the stoppage of growth. So arrested primordial follicles, they are around 40,000 in number. They are around 40,000 in number. So here what happens is at puberty, exactly at puberty, what happens is this arrested meiosis, the process of arrest is lifted up. It is gone. And now there is growth of this primordial follicle into secondary oocyte. And these secondary oocyte, every month, one secondary oocyte is released out. Before this, the formation of Graafian follicle occurs. And this follicle pops out an ovum, the secondary oocyte comes out. And this entire process, we are calling it as a ovarian cycle. The flowchart that you are reading is what? It is the oogenesis. So from the primordial germ cell to oogonia, from oogonia to the primary oocyte. So please remember, from the oogonia to the primary oocyte. So these three stages, from the primordial germ cell to the primary oocyte, this is occurring in the prenatal life. That is embryonic life. Alright, at birth what happens? At birth, it is formation of, so at birth what's happening is formation of primordial follicle is occurring. Primordial follicle is occurring. Please remember that at birth, the primary oocytes are around 7 million in number. Alright, prenatal life means what? First month, second month, third month, fourth month, fifth month, sixth month, seventh month, eighth month and ninth month of pregnancy. From first month to ninth month of pregnancy. So please remember, the formation of oogonia is occurring from second to third month of pregnancy. The formation of primary oocyte begins from third to 4 months. And the primary oocyte, the meiosis is started at 7th month. Alright, the 7th month, 7 millions. You will have to remember this. 7 month, 7 millions. 7 month, 7 millions. Alright, at birth. So after that, the primordial follicles which are arrested, the arrested primordial follicles in the prophase 1 of meiosis, they are around, initially they are around 1 to 2 lakhs. 1 to 2 lakhs in number before puberty. They are around 1 to 2 lakh in number before puberty. You can imagine from 7 million. 7 million means 70 lakhs. It has come to 1 to 2 lakhs before puberty. At birth it was 7 million. Just before puberty it has come to 1 to 2 lakhs. At puberty. So before puberty 1 to 2 lakhs. At puberty we have got 40,000. At puberty we have got 40,000. Each and every point is important in your exams here. They'll ask you what is the number of cells at puberty, what is the number of cells before puberty, what is the number of cells at birth. They'll ask you. At birth 7 million, before puberty 1 to 2 lakhs, at puberty it is 40,000. And throughout the life of a female, every 1 month one oocyte is released, is it not? So like that, around 500 oocytes are necessarily becoming Graafian follicle throughout the life of a female. How is this 500 number? I'll tell you when I am teaching gynecology infertility chapter. But for now, please remember that the ovarian cycle is a cycle in which the primordial follicles are getting converted to secondary oocyte, and it is because of the formation of the mature Graafian follicle. So in the ovarian cycle, what's happening is the meiosis 1 again starts. So the meiosis 1 begins and it continues, starts continuing, and it gets completed. Once it gets completed, the completion of first meiotic division, results in formation of what is known as secondary oocyte. Secondary oocyte is formed The rest of the cell we are calling it as a polar body. So one secondary oocyte and one polar body. We know that at the end of meiosis 1, one cell divides into two daughter cells. Each will have haploid number of chromosomes. So here we have got 22X and here also we have got 22X. I'll ask you to recall what was the yes. Yes, here I wrote. So what was the chromosome number in the oogonia? I want you to recall this. It was 44XX. Now if you see at the end of meiosis 1, it has become 22X in a secondary oocyte and 22X in the polar body. 22X in the secondary oocyte and 22X in the polar body. At the time of puberty, they have come down to around 500 secondary oocytes and polar bodies. Around 500 secondary oocytes and polar bodies. So after this, what happens? After this, again, the second meiotic division begins. Second meiotic division begins. The second meiotic division also undergoes arrest in metaphase. Why it is undergoing arrest in metaphase? Because of every month the fertilization does not occur, is it not? Only if the fertilization occurs, the second meiotic division completes. For fertilization to occur, sperm should be present in the tract. For sperm to be present, reproduction should have happened. Reproduction is a process which occasionally happens, not at puberty, beyond that maybe at some time. So every month the secondary oocyte is released and it is wasted like that. If at all fertilization is about to happen, what happens is the ovum combines, the secondary oocyte is ovum. The secondary oocyte is ovum. So the ovum combines with the spermatozoon to form a zygote. And if fertilization is occurring, this fertilization results in completion of the second meiotic division. This results in completion of second meiotic division. That is with formation of zygote. All right, with formation of zygote, right? So after ovarian cycle, the second meiotic division that is occurring, it is occurring in the phase of ovulation. Ovulation. And second meiotic division undergoes arrest in metaphase. It has to get completed in fertilization phase. So please remember, right from the prenatal life, I'll highlight this with yellow color, right from the prenatal life, till the birth, it is different. Immediately following birth, before puberty, it is different. 1 to 2 lakhs of of the primordial follicles. At puberty, we have got around 40,000 follicles, and there is a arrest of meiosis already occurred. The arrest of meiosis has occurred before puberty itself, is it not? Arrest of meiosis occurred before puberty itself. And then what happened was ovarian cycle has started. Every month around one oocyte is released. Meiosis 1 starts continuing. It was arrested in the prophase 1 or diplotin stage of prophase 1. I'll write somewhere here. So the meiosis 1 arrested in the, so arrest of meiosis 1 has occurred when? Before puberty. In which stage? The diplotin stage of what? Of prophase 1. The meiosis 2 got arrested when? At the time of ovulation. Every month it is happening. And in which stage? In the metaphase 2 stage. You have to understand this. Lot of MCQs are asked. Very, very important concept to understand, especially the infertility treatment. Infertility treatment especially, right? So to summarize, what happens is the primordial germ cells, or the primitive sex cells during the 4th week of development. So I repeat again, the primordial germ cells in the 4th week of development is 1st month. So they appear in the wall of the yolk sac. They appear in the yolk sac in around 4 weeks of development. 4 weeks of development. The primordial germ cells undergo mitotic divisions. They undergo mitosis. During their migration, they undergo mitosis and result in formation of the oogonia. So please remember that this process, the process of maturation and differentiation of primordial germ cell to oogonia, from oogonia to primary oocyte, from primary oocyte to primordial follicle, from primordial follicle to Graafian follicle, from Graafian follicle to secondary oocyte, to the secondary oocyte to mature ova in the female genital tract. This entire process is called as oogenesis. Where is it occurring? It is occurring in the ovaries. When it is starting before birth. It stops at birth. At birth there is first break to it, the meiosis 1. Then what happens? It restarts at puberty. At puberty it is restarting it. Arrest of meiosis is gone. Meiosis 1 continues at puberty. It continues like that and again stops in the middle before fertilization. If fertilization occurs, it continues and then it completes the meiosis 2. Please remember, in the process of oogenesis, what are the important steps that are occurring? First mitosis, then meiosis. First growth of the follicle, then differentiation of the follicle, and ovulation. Ovulation is a very, very important step that is occurring because of changes in the hormone levels. The oogenesis in different phases of life, that is before birth. So before birth, that is prenatal life. What's happening here? Before birth, the primordial germ cells are undergoing mitosis to form oogonia. This occurs because of absence of what is known as the testis differentiating factor. This is a growth factor. If it is absent, this results in the oogenesis, that is change of primordial germ cell, differentiation of primordial germ cell to form oogonia, and mitosis of the primordial germ cell to form oogonia. All right, hope you have understood this till now. So before 7th month is an important parameter here. 7th month, I told you 7th month is such an important phase here that just before 7th month, what happens is the oogonia is getting continued to divide mitotically and to form primary oocytes. At 7 months, from 7 months till birth, what's happening is the primordial follicle is formed. The primary oocyte undergoes changes to form primordial follicle. The primary oocyte now starts undergoing meiosis 1. But suddenly what happens is in meiosis 1, what is the first stage of meiosis 1? Prophase 1. What are the important stages of prophase 1? Yesterday I told you. So in that diplotin phase, prophase 1, there is a arrest of meiosis 1. This arrest of meiosis 1, is brought about by an important inhibitory factor called as oocyte maturation inhibitor. Oocyte maturation inhibitor. The oocyte maturation inhibitor is responsible for the formation of the arrest of meiosis 1. It is something like this. So what happens is oogonium. Say suppose this is the oogonium. What is oogonium? 44XX, is it not? It undergoes change into a primary oocyte. by mitosis. So 44x, all right, double X. Then what happens is the primary oocyte undergoes first meiotic division. In this process, one polar body, the first polar body, is released. So the first polar body has got 22x and secondary oocyte is formed. This secondary oocyte has 22x. This is meiosis 1. This is meiosis 1. From here, the secondary oocyte undergoes second meiotic division, meiosis 2. But here also arrest occurs in the metaphase. And from here what happens is, if at all it continues, then formation of second polar body occurs at fertilization. Formation of second polar body occurs at fertilization and formation of ovum. So this is the secondary oocyte, or this is the after secondary oocyte, the maturation of secondary oocyte. From the secondary oocyte, if you remove the second polar body, it results in formation of ovum. So ovum is 22x and this is also 22, polar body is 22x. So they'll ask you important question. First polar body is formed at the time of ovulation. Second polar body is formed at the time of fertilization. Please remember this point. First polar body is formed at the time of ovulation. Second polar body is formed at the time of the fertilization. Please remember that the oogonia are diploid, initially diploid chromosome, oogonia. So the oogonium is diploid, the primary oocyte is also diploid, but the secondary oocyte is haploid. You will have to remember this point. So we are discussing what we have already discussed from first few months, that is up to 7 months we have discussed, is it not? So what happens from birth to before puberty, just before puberty, what happens? Both maturation and differentiation of the primordial follicle occurs, but the problem is here meiosis is arrested. In the first prophase 1 of first meiotic division, their arrest occurs. Around 2 lakhs primordial follicles are present at birth. All right, excuse me. So next starts the puberty. At the time of puberty, what happens is the ovarian cycle starts. So at this time we have got around 40,000 in number. The primordial follicles are 40,000 in number. The primordial follicle starts continuing with the meiosis 1. Ovulation takes place. Ovulation takes place in this phase. Every month ovulation takes place and one oocyte is released. So the secondary oocyte immediately enters into the second meiotic division. So with release of first polar body, it results in formation of the secondary oocyte. So the primary follicle, so where did I draw this? All right. So with this, what happens is the second meiotic division is completed only if fertilization occurs. The second meiotic division occurs completed only if fertilization occurs. So time has come now to understand the importance of reproductive phase. In the reproductive phase, there are 400 primordial germ cells. Out of 40,000 primordial germ cells, I'll talk about numbers now. 40,000 to only 400 to 500 primordial germ cells, they release the ovum. They release the ovum. All right. And out of these, only 3 or 4 or whatever it is the number, if they undergo fertilization, results in formation of a fetus. All right. So in this, you will have to understand that what was the changing point here. The ovarian cycle was a turning point actually. So we'll try to understand ovarian cycle. We'll try to understand ovarian cycle. The ovarian cycle is defined as, it is the cyclical change that is occurring in the ovary during 28 days of reproductive cycle. cyclical changes that occur in the ovary, cyclical changes that occur in the ovary where in the ovary in the 28 days of reproductive cycle or the menstrual cycle. So here what happens, an important step of ovarian cycle is release of ovum. The process is called as ovulation. The process is called as ovulation. The process is called as ovulation. It is under the influence of anterior pituitary hormones, that is the FSH and the LH. LH surge, I told you in my physiology class of endocrinology, I told you LH surge was responsible for ovulation. All right. So initially what happens, the primordial follicles, they grow. The primordial follicle grows into primary follicle into a mature primary follicle called as Graafian follicle. This Graafian follicle has got a follicular cavity called as antrum surrounded by cells. It undergoes changes into secondary follicle after releasing the ovum, and a mature Graafian follicle, it looks something like this. All right. So let us try to understand the follicular genesis of ovarian cycle. Let us try to understand this point now. So follicular genesis. We have got the primordial follicle. We have got the primordial follicle. This primordial follicle, what happens is the oogonia. We all know that oogonia gives rise to primordial follicle. It changes itself into primordial follicle because it is surrounded by the matrix cells. It is surrounded by the stromal cells. Some of the stromal cells which are surrounding the oogonium or oocyte, they actually become flattened and surround it to form a primary oocyte. They ultimately make it get converted to follicular cells. For example, so this is the ovum, or this is the primordial follicle, and the cells which are present around, they unite, they get flattened like this to surround the primary follicle. And now these cells are forming the boundaries of the ovum, the future ovum, whatever it is, the primordial follicle, the primary oocyte. Now each primary oocyte is covered by a single layer of flattened follicular cells. This is called as primordial follicle. This is called as primordial follicle. Primary oocyte surrounded by flat cells, follicular cells. You are calling it as a primordial follicle. Both the primary oocyte and the granulosa cells are covered by a thin basement membrane. So what is the granular cell? Let us try to understand. Before that, try to understand that the primordial follicles, they are the basic reproductive units at puberty. Especially at puberty, this is happening. All right. They are seen in the deeper cortex of the ovary. All right. From the 10th week of development till the puberty, this is what is happening. So at birth, approximately 2 lakh primary oocytes are present. I told you this. At birth, 2 lakhs primary oocytes are present. All right. They undergo degeneration subsequently. The number of primary oocytes is reducing, and at puberty, it is 40,000. At puberty, it is 40,000. From birth to puberty, it is reduced to 40,000. And from just before puberty, I'm talking about just before puberty. So I'll go back to this. Statement here. So just before puberty, it was 2 lakhs. At puberty, it has become 40,000. At puberty, it has become 40,000. So 400 to 500 mature. Out of these 40,000, only 500 they mature. Only 500 Graafian follicles are formed. That is, they mature to release an ovum or to undergo a process of ovulation. So this is what you need to understand. From here, only 500 are required, which become Graafian follicle. What is Graafian follicle? Now let us try to understand. The primary follicles, the oocyte which is completely surrounded by the cells, it undergoes first meiotic division. So from puberty to menopause, during the reproductive phase of a female, what happens is, under the influence of FSH and LH, several primordial follicles, they start maturing each month. Each month what happens is follicular stimulating hormone, the FSH, so in the ovary. So this is the ovary. We have got multiple primordial germ cells, primordial follicles. Out of these, what happens is around 10 to 50 cells are selected by this FSH. This FSH is coming from the pituitary gland, if you remember. Pituitary is releasing FSH. FSH is acting on the ovaries. Who is controlling pituitary? The hypothalamus by GnRH. GnRH. So this is termed as the hypothalamus pituitary ovarian axis. So what happens here is, the follicular stimulating hormone, it selects around 10 to 50 follicles. You, all of you, every month. Now this month you are the persons who are selected. It will say, I'm just correlating it like that. So follicular stimulating hormone will come down to the ovary and say, I am selecting 10 to 50 follicles. Out of these 10 to 50, all of you start growing it. All of you start growing yourself, and out of you the mature Graafian follicle will be selected, and this Graafian follicle has to have the property of ovulation. This is occurring every month. All right. So what's happening here is, these cells will undergo maturation and form a larger follicle. All right. Other follicles are suppressed. The follicular cells undergo mitotic division to form several layers of follicular cells, layer after layer, layer after layer. They are increasing. So I have drawn this diagram. No. This undergoes multiple divisions to form layers like this, layers of cells. Multiple layers of cells like this. All right, layer by layer. This we are calling it as a multilaminar or multi-layered follicle. This multi-layered follicle is surrounding the primary oocyte. So we have got the primary oocyte here, and it's a multi-layered follicle. It's a multi-layered follicle. All right. This primary oocyte, it increases in size. The follicular cells now are called as granulosa cells. These follicular cells are now called as granulosa cells. These follicular cells are called as granulosa cells. Now in this granulosa cell, what happens is, a small cavity is formed. A small cavity develops, fluid filled cavity develops. All right. A fluid filled cavity develops. Cells surrounded by a fluid filled cavity, we are calling it as a cyst or a follicle. Actually, these cells got a name of the follicle because of this property of antral fluid. So antrum means empty space. Antral fluid, any fluid which is surrounded by cells, you are calling it as cyst. So this is the antral follicle. The growth of the follicle, what happens is, the fluid spaces in between the granulosa cells, they coalesce to form a bigger follicle, bigger follicular antrum. And as the growth occurs around the 7th day of sexual cycle, this is occurring at the 7th day of the sexual cycle, under the influence of follicular stimulating hormone, under the influence of LH, what's happening is, the Graafian follicle, a fully matured Graafian follicle is formed. This Graafian follicle, so if this is the ovary, Graafian follicle, somewhere here it reaches the peripheries of the ovary. All right, I'm not drawing it exactly, But I'm just showing you, I want to show you that there is a oocyte which is surrounded by cells. All right, and it goes to the periphery. It is because of the fluid, it gets pushed to the periphery, and one fine day what happens is it has to release this primary oocyte outside. This process we are calling it as ovulation. The layers which are surrounding the Graafian follicle, the layers which are surrounding immediately surrounding the oocyte, you are calling it as a zona pellucida. These are the layer of cells which are surrounding the primary oocyte. The primary oocyte and zona pellucida together we are calling it as a corona radiata. Corona means crown like, corona radiata. The projection of these granulosa cells into the follicular antrum forms what is known as cumulus oophoricus. So we have got zona pellucida. So if this is the oocyte, it is surrounded by cells. We are calling it as zona pellucida. And this entire structure we are calling it as a corona radiata. We have got a follicular cavity like this, and these cells which are projecting into the follicular cavity eccentrically, not in the center, eccentrically, you are calling it as a cumulus oophoricus. Cumulus oophoricus. As the follicle expands, the stromal cells surrounding the membrana granulosa, the area of attachment of primary oocyte and corona radiata to the wall of the follicle is called as discus proligerus. I'll tell you what is that. So if this is the follicle, these are the cells of the follicle. Another layer, another layer. All right. This is the fluid filled space. Yellow color is the fluid. All right. The area of the primary oocyte, this area, these cells on which the primary oocyte is sitting, the area of the Graafian follicle on which the primary oocyte is attached to the Graafian follicle, you are calling it as discus proligerus. Discus proligerus. Discus proligerus. So too many different names. I repeat, zona pellucida is the cells which are surrounding the primary oocyte. The entire structure we are calling as corona radiata. It is forming a projection into the antrum. We are calling it as cumulus oophoricus. The primary oocyte sits on few cells on the Graafian follicle. You are calling it as a discus proligerus. As the follicle expands, the stromal cells surrounding the membrana granulosa, they become condensed to form theca interna. Theca interna. And the theca interna, what is the specialization of theca interna is, it is increasing in thickness, it becomes more vascular. So the it has got a lot of vascularity, hence theca interna is a place which is secreting the estrogen. Because of the presence of vasculature. Outside the theca interna, we have fibrous tissue which is covering another follicle. We are calling it as theca externa. Theca means covering. Externa means external. Theca externa. The follicle gradually increases in size and finally it pops out an ovum, and the process we are calling it as ovulation. Just before ovulation, what happens? The primary oocyte of the Graafian follicle completes its first meiotic division to form secondary oocyte and a polar body. Secondary oocyte and a polar. Now the secondary oocyte gets transformed into the ovum, secondary oocyte and the first polar body. Secondary oocyte and the first polar body. Please remember that this is all about the formation and the structure of the Graafian follicle. All right. So mature Graafian follicle, I'll again draw it separately for you. Say suppose if this is the primary oocyte, so pay attention now. I'm just revising what I told you. All right. We have got cells which are surrounding the primary oocyte. The cells which are surrounding the primary oocyte you are calling it as a zona pellucida. Zona pellucida. All right. This entire structure you are calling it as what? Corona radiata. And it is projecting, these cells are projecting into the antrum cavity. All right. And these cells are projecting into the antrum cavity. And we are calling it as the cumulus euphoricus. These cells are surrounded by the membrana granulosa cells. So now we have got membrana granulosa cells. This is the antrum cavity. Membrana granulosa cells. The area of membrana cells, the pink color here is membrana granulosa cells. The area of membrana cells on which the primary oocyte is sitting eccentrically, you are calling it as discus proligerus. Now there is a thin and thick layer of cells surrounding this. I'll draw antrum cavity now. So this is full of fluid which is rich in FSH and LH. And now this is surrounded by important lining. This is surrounded by an important lining called as theca interna. It is surrounded by again an important lining called as theca externa. This is called as a mature Graafian follicle. So centermost portion we have got the oocyte. Immediately following oocyte we have got the zona pellucida cells, and this portion you are calling it as cumulus euphoricus. This portion on which the cumulus euphoricus sits we are calling it as discus proligerus. We have got the antrum. These are the membrana granulosa cells. Pink color cells are membrana granulosa cells. This is all about the mature Graafian follicle. This is all about the mature Graafian follicle. Now, ovulation results in shedding of the primary oocyte, now which is called as secondary oocyte, into the cavity, into the abdominal cavity, from where it is picked up by the fallopian tubes. And if at all in the fallopian tubes the sperm is present, fertilization occurs. So quickly we'll try to understand what is the sequence of events of that are occurring during ovulation. So what happens is, because of LH surge, this Graafian follicle pops out the ovum. The centermost, this eccentrically present ovum is popped out. Once the ovum comes out, Graafian follicle becomes corpus luteum. Luteum means yellow. Corpus means mass of cells. And it starts secreting progesterone. which prepares the uterus for pregnancy. Immediately following ovulation, every month the uterus is expecting pregnancy to occur. Hence, progesterone hormone is important. It is released. It is secreted by the corpus luteum cells of the corpus luteum, which is nothing but Graafian follicle devoid of ovum. Graafian follicle after ovulation, it is called as corpus luteum. All right, corpus luteum. The follicular cells are showing increased activity. There is loosening of cumulus cells. Ischemia occurs at the place of discus proligerus. Discus proligerus ischemia occurs, and ovulation occurs due to high concentration of LH just before ovulation. Around 72 hours after LH surge, ovulation occurs. All right, please remember this point. So this is all about the oogenesis and release of the ovum. Menstrual cycle I already discussed in my endocrinology class. I'll again discuss menstrual cycle in gynecology chapter. Gynecology chapter I'll again discuss menstrual cycle. All right. Now we'll discuss a few important points about sperms. We'll try to understand few important points about sperms. Spermatogenesis I already discussed clearly. I discussed what are the different steps that are occurring in spermatogenesis. So stages of spermatogenesis, like from the primordial germ cell to the spermatogonia, from spermatogonia to the primary spermatocyte. From the primary spermatocyte undergoes first meiotic division to form secondary spermatocytes. Secondary spermatocyte becomes spermatids. Spermatids undergo morphological changes to become spermatozoa. The last step we are calling it spermiogenesis. Spermiogenesis. So the spermatid is more or less a round cell. Spermatid is a round cell, whereas spermatozoa, it has got a head, a neck, tail. It looks something like this. It has got a head, neck, and it has got a tail like this with a nucleus. Spermatid is a round cell with a nucleus. So drastic changes are occurring from spermatid to spermatozoa. This process is spermiogenesis. We'll try to understand what is spermiogenesis now. So in case of spermiogenesis, what happens is there is nuclear condensation first. Nuclear condensation, then formation of tail. Nuclear condensation, then formation of a small tail, then formation of acrosome or head. Acrosome we call it. Acrosome of the sperm. Formation of acrosome. Then rearrangement of cells like mitochondria and organelle, various cell organelles are arranged like this. So we'll try to draw a sperm and we'll try to understand how is the sperm looks like. So we have got this is the head of the sperm. Then we have got the neck constriction. Then we have got the middle piece like this. That is the body. Then the body terminates like this. Then we have got a tail. I'm just turning this tail like this so that you understand it. Alright, this tail is the axial filament which grows outside the centriole. Now what is the centriole, what is the acrosome, we'll try to understand now. So head of the sperm has got Golgi apparatus which forms the cap of the acrosome. It has got a nucleus. So this is the nucleus of the sperm. So this is the nucleus of the sperm. Over over the nucleus of the sperm sits a very important structure. All right, over the nucleus of the sperm sits a very important structure called as acrosome cap. So this is cap. This cap is composed of Golgi apparatus. Golgi apparatus. It's a very, very important MCQ. This is the nucleus. This is the nucleus. I'm sorry. This is the nucleus, and in this area we have got an important structure called as primary centriole. Primary centriole. In this place we have got primary centriole. These are all the cell organelles, or proximal centriole. It is also called as proximal centriole that lies in the neck. From the proximal centriole, the body of the sperm starts and it ends with what is known as distal centriole. Here we have got is it visible? Distal centriole. Yeah, now it is visible. Yes, now it's okay. All right, so in the middle piece or body we have got arrangement of the mitochondria like this. Mitochondria are formed like this. Mitochondria are arranged in the middle piece. So this part is the head, this is the neck, this is the middle piece, this is the middle piece, and the tail is called as the principal piece. So we have got what is known as an axial, axial filament that runs through the from the proximal centriole to the distal centriole like this. This axial filament, it has got a unique property. It helps in motility of the sperm. This axial filament also present in the tail like this. It runs in the tail like this. This we are calling it as axial filament. So Golgi apparatus is present in the cap. Mitochondria are present in the middle piece of the sperm. So this is what I want you to understand. This is a structure of a sperm. So from a round spermatid, we have got first nuclear condensation, formation of tail, formation of acrosome. Then comes the centrosome of the middle piece. The centriole divides into two parts, the proximal centriole and the distal centriole. The proximal centriole lies in the neck, the distal centriole lies in the beginning of the tail. The axial filament grows out of this centriole and it runs in the middle piece. And the distal end of the axial filament, what is formed is the distal centriole. All right. So the cell membrane of this sperm, it has got it recognizes the egg, it binds to the egg, it fuses with the egg. There are three important properties of this cell membrane. It recognizes the egg, it binds to the egg, and it fuses with it, gets reacted with it, it enters into the ovum, and fertilization occurs. Please remember that a mature spermatozoa, it takes lot of process like this to form. The axial filament which passes through the middle piece, it extends up to the tail, and this is helping in the motility of the sperm. If you see the head of the sperm, it is pyriform in shape. It is derived from the nucleus. It consists of 23 chromosomes. And this it has got a cap which is composed of the acrosome cap. We call it acrosome cap, is composed of the Golgi apparatus. The neck is narrow, it is funnel shaped, and it holds the spherical proximal centriole. From the centriole, the axial filament enters into the middle piece or the principal piece. In the middle piece, we have got peripherally arranged mitochondria and central axial filament. All right. So if you take a transverse section of this principal piece or tail looks something like this on cross section. On cross section, the tail looks something like this. We have got coarse petals in the tail. We have got filaments or fibrous sheath. So these are the doublets. We call it doublets. These doublets will give rise to filaments like this. All right. These are the doublet cells. These are doublets, and we have got coarse petals like this. This is a cross section of a tail, and it is surrounded by the fibrous sheath like this. So this is the fibrous sheath. These are the doublet cells, and this is the coarse petal. All right. And this is the plasma membrane. All right, this is the structure of an axial filament. This is the structure of the axial filament, which is composed of the central fibrils. So this is the central fibril, and it is attached to the peripheral fibrils like a flower, like a so we call it doublets. We have got 9 pairs actually, 9 pairs of doublets, and there is a central pair, so total 10. So total 9 pairs like this. So 1 pair, 2 pair, 3 pair, 4th pair, 5th pair, 6th pair, 7th pair, 8th pair, and we have got 9th pair like this. So 9 peripheral pairs of doublets and 7 central pair. The whole system of fibril is put in place with the help of these coverings of fibrous sheath. In the middle piece, the fibrous sheath is surrounded by the mitochondria. In the tail, we have got the axial filament continuing, and this is responsible for movement of a spermatozoa. All right, and this is surrounded by a plasma membrane. Plasma membrane also has got important function. Plasma membrane also has got important function in in the proliferation of sperms. So this is all about. So we have come to a conclusion of the topic. This is all about the spermatogenesis and oogenesis. Hope you have followed the class. In my discussion, if you could not follow anything, you ask me. I'll just repeat the topic again. Thank you very much for patient listening.